Hello students, you have studied structure of nephron in the previous class. Today we will study the different type of nephron and further the process of urine formation. So there is a quick revision of the structure of nephron. That nephron is made up of the glomerulus and renal tubule. Glomerulus is the structure which is formed by the efferent arteriole, which is a fine branch of renal artery, and glomerular capillary. They unite to form the efferent arteriole. Then we will come to the renal tubule. So this is the renal tubule. Renal tubule is further made up of the Bowman's capsule, PCT, loop of Henle, and then the DCT. So Bowman's capsule is the double-walled cup-shaped structure, and the two walls of Bowman's capsule here are inner visceral and outer parietal, and both are single layered and are supported over the basement membrane. Then we will come to the Malpighian body. Malpighian body is actually glomerulus along with the Bowman's capsule is termed as the Malpighian body or renal capsule. So together they are known as the Malpighian body, this glomerulus and Bowman's capsule. Then the PCT is there, the proximal convoluted tubule, lower part, and this is present in the cortex, highly coiled, surrounded by peritubular blood capillaries lined by cuboidal epithelium, having brush borders with long microvilli for increasing absorptive area. Then the loop of Henle. This is the loop of Henle. Loop of Henle, it is a hairpin loop-like tubular part of nephron which descends into the renal medulla, and this is made up of two parallel limbs joined by the curved base. There is descending limb and ascending limb. So this is the descending limb, this is ascending limb. Then next we will come to the distal convoluted tubule. Distal convoluted tubule is actually highly coiled part of the nephron, and it lies very closely to the Bowman's capsule or Malpighian body. Then we will come to the collecting duct. Collecting duct actually this is the this is receiving that DCTs of many nephrons. So the DCTs of many nephrons open into a straight tube. This straight tube, it is termed as the collecting duct, many of which converge and open into the renal pelvis through medullary pyramid in the calyces. So this collecting duct, it opens into the renal pelvis through the medullary pyramids. Then next we will see the Vasa recta. This is the structure of the Malpighian body. Here these are the efferent arteriole. This is the efferent arteriole. This is Bowman's capsule, the cup shape, that cavity here. Together they both are termed as the Malpighian body. This is glomerulus and Bowman's capsule together they termed as the Malpighian body. Then the next we will come to the types of nephron. In majority of nephrons, the loop of Henle is too short and extend only very little into the medulla. Such nephrons are called as the cortical nephrons. So cortical nephrons have the short loop of Henle, and very little they are extended into the medullary region. So they are termed as the cortical nephron. In some of the nephron, the loop of Henle is very long. Like here you can see that the loop of Henle is being very long and runs deep into the medullary region. They are known as the juxtamedullary nephron. So these are the juxtamedullary nephron which have long loop of Henle, and cortical nephron, those who have the short loop of Henle. Maximum their parts are located in the cortical region. Now Vasa recta, the efferent arteriole emerging from the glomerulus forms a fine capillary network around the renal tubule. These tubule together, they termed as the peritubular capillaries. These that capillaries actually they call the peritubular capillaries. A minute vessel of this network runs parallel to the Henle's loop, forming a U-shaped vasa recta. Vasa recta is absent or highly reduced in the cortical nephron. So vasa recta is also the network of the vessels. So minute vessels of this network runs parallel to the loop of Henle and forming that U shape, that is the vasa recta. So peritubular capillaries, the network of the capillary around the nephron, and vasa recta, it is the network of the vessel running parallel to the Henle's loop. So this is about the vasa recta. Now we will come to the physiology of urine formation. So physiology of urine formation involves 3 steps. 3 main processes are there, namely the glomerular filtration, reabsorption, and secretion. 3 processes are there that takes place in the different parts of the nephron. So the first step, this is the filtration. Filtration of the blood, and then after this filtrate is carried to the different parts of the loop of Henle. So this filtration is taking place in the Malpighian body here, in the glomerulus and Bowman's capsule, both are involved. Then the filtrate, it travels through the renal tubular part of the nephron, means renal tubule, where the reabsorption of the beneficial substances takes place, and the waste substances, they are secreted in the tubule. So we will study the all 3 processes here. So glomerular filtration is the first step in the urine formation. Here where the filtration of the blood is taking place, which is carried out by the glomerulus, and it is called as the glomerular filtration also. On an average, 1100 to 1200 ml of blood is filtered by the kidneys per minute, which constitute roughly 1/5 of the blood pumped out by each ventricle of the heart in a minute. The glomerular capillary blood pressure causes filtration of blood through 3 layers. So a reason for the filtration is the capillary blood pressure. Which one? The glomerular capillary blood pressure. And this filtration is taking place through the 3 layers. These are the endothelium of glomerular blood vessels, the epithelium of the Bowman's capsule, and the basement membrane between the 2 layers. The epithelial cells of the Bowman's capsule, called podocytes, are arranged in intricate manner so as to leave some minute spaces called filtration slits or slit pores. So in this manner they help in the filtration. And blood is filtered so finely through these membrane that almost all the constituents of the plasma, except the proteins, pass into the lumen of Bowman's capsule. Therefore, it is considered as the process of ultrafiltration also. And the amount of filtrate formed by the kidney per minute is called glomerular filtration rate, that is GFR. And GFR in healthy individual is approximately 125 ml per minute, that is 180 liters per day. So the development of the filtration, the pressure is due to the glomerular capillaries. Why it is so? There are 2 reasons. That the larger diameter of the efferent arteriole as compared to the smaller diameter of the efferent arteriole. So this is actually playing the important role. So efferent arteriole and efferent arteriole, both they have the different diameter. The diameter of the efferent arteriole is being the larger one. This is efferent arteriole. and this is the efferent arteriole. So efferent arteriole, they have the smaller diameter. So here, this is the reason which causes the pressure here in the glomerulus, and natural arterial pressure is caused by the pumping activity of heart. Blood pressure in glomerular blood is about 60 mmHg. It is about 60 mmHg, and this is known as the glomerular hydrostatic pressure, that we call it as the GHP, glomerular hydrostatic pressure. Osmotic concentration of proteinaceous content of glomerular blood is equivalent to 30 mmHg, and this is called blood colloidal osmotic pressure. So this is the pressure of glomerular blood. Here due to the proteinaceous content. Now the pressure of the interstitial fluid and pressure of the renal filtrate is collectively known as the capsular hydrostatic pressure, that is CHP, and this is 20 mmHg. So the pressure being exerted on glomerular blood for undergoing filtration is called the glomerular filtration pressure, GFP, which is about 10 to 25 mmHg. So in this manner, this pressure is responsible for the filtration of the blood through the glomerulus. Now we'll see the glomerular filtration here. So as I said you, this is the Bowman's capsule here. I explained you these are the podocytes and the inner spaces in between them through which that blood passes, and these are the mesangial cells, efferent arteriole. This is efferent arteriole. So difference here in the diameter of the efferent arteriole and the efferent arteriole. Here the diameter of the efferent arteriole is more than the efferent arteriole. So more blood enters into the glomerular capillaries, and when it goes, then the amount of blood goes, it will be less because of the less diameter. So here in the glomerular filtration, the three methods are there by which renal blood flow and GFR, that is the glomerular filtration rate, are regulated. First, the myogenic autoregulation. Second is juxtaglomerular apparatus, and third is the neural control. तो myogenic autoregulation में क्या होता है? A rise in blood pressure should normally increase the blood flow through the glomeruli. तो जैसे ब्लड प्रेशर राइज होगा तो ब्लड का फ्लो जो है वो इंक्रीज होगा ग्लोमेरुलस में. However, stretching of the vascular wall increases passage for the calcium ions from extracellular fluids into the cells, resulting in the contraction, and contraction checks the overstretching of vascular walls and raised vascular resistance so that the blood flow and the GFR are brought down to the normal. तो ये है myogenic autoregulation. Juxtaglomerular apparatus का क्या रोल होता है? That is known as the JGA also. So JGA is a spatial sensitive region formed by cellular modification in DCT and the efferent arteriole at the location of their contact. So JGA become active when there is decrease in the renal blood pressure or decrease in GFR. So this JGA promotes release of renin. This is the hormone renin from the juxtaglomerular cells, and this renin converts protein angiotensinogen into peptide angiotensin, and angiotensin is also hormone that raises the glomerular blood pressure through constricting efferent arterioles, resulting in restoring GFR. Third है neural control, where the blood vessels of the kidney are innervated by the nerve fibers of sympathetic nervous system, and when activated, the nerve fibers bring about constriction of the renal arteries and causes decrease in the renal blood flow as well as glomerular filtration rate. So this is the regulation of the glomerular filtration. Now after this glomerular filtration, next we will come to the tubular reabsorption. So here also in this diagram you will see the efferent arteriole and efferent arteriole. They have the different diameter here. Here the diameter is higher, here the diameter is less. So more blood enters into the glomerulus here, then it goes out here, it exerts the pressure here, and that causes the filtration. And here you can see how the filtration is taking place. That blood is entering into the glomerulus, then it is undergoing filtration, and after filtration blood is entering into the efferent arteriole, and further the filtrate is also further passing through the PCT, and then the loop of Henle, and then the DCT. Now we will come to the tubular reabsorption. Tubular reabsorption is that process here in which the reabsorption of the important substances takes place in order to maintain the concentration of blood. This is known as the selective reabsorption also, because in selective reabsorption sodium, amino acid and glucose, they are reabsorbed from the filtrate back into the blood. So this is the passage where the reabsorption takes place. So reabsorption takes place in PCT, then the loop of Henle, and then the DCT. So we will study here what happens in the tubular reabsorption. A comparison of the volume of filtrate formed per day, that is 180 liters per day, with the urine released suggests that nearly 99% of the filtrate has to be reabsorbed by the renal tubule. तो जो 99% जो पार्ट होता है फिल्ट्रेट का, वो 99% पार्ट जो है वो रीएब्सॉर्ब्ड हो जाता है, मतलब 1% जो है वो वेस्ट प्रोडक्ट की तरह एक्सक्रीट होता है. This process is called the reabsorption, and the tubular epithelial cells in different segments of nephron perform this either by active or passive mechanism. तो इसमें दोनों mechanism यूज होते हैं, active mechanism as well as passive mechanism. For example, substances like glucose, amino acids and sodium, they are absorbed in the filtrate actively, मतलब active absorption के through होते हैं, whereas the nitrogenous waste, they are absorbed by passive transport. Reabsorption of water also occurs passively in the initial segment of the nephron. So here you can see the absorption of that chloride, water, nutrients, potassium, it is taking place here, and water is also reabsorbed here. And here you can see the urea secretion में क्या होगा, urea, ammonia, they are secreted into the filtrate. तो ट्यूबुलर सिक्रीशन के अंदर क्या होगा, द फिनोमिनन ऑफ सिक्रीशन ऑफ मेटाबॉलिक वेस्ट बाय ट्यूबुलर वॉल्स इनटू द फिल्ट्रेट. तो ये जो मेटाबॉलिक वेस्ट हैं, यूरिया एंड अमोनिया, दे आर एक्चुअली सिक्रीटेड इनटू द filtrate. This is known as the tubular secretion. It includes the secretion of potassium ion also, and hydrogen ion, ammonia, some waste like hippuric acid, creatinine, drugs, pigments, toxins, and it is only the mode of excretion in animals which do not have glomeruli. For example, desert amphibians, marine fishes. Tubular secretion is also an important step in the urine formation, as it helps in the maintenance of ionic and acid-base balance of the body fluids. So now one by one we will see here that the role of the PCT, loop of Henle and DCT as well as the collecting duct in the process of urine formation. तो PCT में क्या होगा, जो PCT है, this is the proximal convoluted tubule. It is lined by simple cuboidal brush border epithelium, which increases the surface area for the reabsorption, and nearly all the essential nutrient, that is 70 to 80% of electrolytes and waters, they are absorbed in this segment. PCT also helps to maintain the pH and ionic balance of the body fluids by the selective secretion of the hydrogen ions and ammonia into the filtrate and by absorption of the bicarbonate from it. So absorption of bicarbonate is taking place here, and the secretion of the hydrogen ion and the ammonia takes place through the PCT. Then we will come to the loop of Henle. So in the loop of Henle, you will see the two limbs are there, the descending limb of loop of Henle and the ascending limb of loop of Henle. So reabsorption is minimum in the ascending loop of Henle. However, this region plays a significant role in maintenance of high osmolarity of medullary interstitial fluid, क्योंकि ये medulla में located होता है. So it plays an important role in the maintenance of high osmolarity of medullary region. The descending limb of loop of Henle is permeable to the water. This is descending limb. It is permeable to the water. So here the water that reabsorption takes place here, but it is impermeable to the electrolytes. So descending limb allows the reabsorption of water, but as it is being impermeable to electrolytes, so no electrolytes passes through it. But in the next that part, the ascending loop of Henle, it is permeable. Here it is showing the different that nature, that ascending limb is impermeable to the water but allows the transport of electrolytes actively or passively. तो यहाँ पर जो electrolytes हैं वो actively जो है वो reabsorb होते हैं, but water जो है यहाँ पर reabsorb नहीं होता है. And this concentrates the filtrate as it moves down. Therefore, as the concentrated filtrate pass upward, it gets diluted due to the passage of electrolytes through the medullary fluid. तो यहाँ पर जो है फर्स्ट पार्ट में जो डिसेंडिंग लूप ऑफ हैनले, यहाँ पर वाटर रीएब्सॉर्ब होता है, तो जो फिल्ट्रेट है वो कंसंट्रेट हो जाता है, एंड देन इट मूव्स डाउन, and then the ascending limb which is impermeable to water but allow the transport of electrolyte actively और passively दोनों तरीके से. तो electrolyte जो है वो जो है ट्रांसपोर्ट होते हैं इस लिम्ब के थ्रू. So the concentrated filtrate passes upward and it gets diluted due to the passage of electrolytes through the medullary fluid. तो electrolyte जो है वो यहाँ पर reabsorb होते हैं, तो वापस से इसकी जो concentration है वो जो है मेंटेन रहती है. डिस्टल कॉन्वोल्यूटेड ट्यूब्यूल में क्या अब हम डिस्कस करते हैं. ये है डिस्टल कॉन्वोल्यूटेड ट्यूब्यूल. डिस्टल कन्वोल्यूटेड ट्यूब्यूल में क्या होता है, कंडीशनल रीएब्जॉर्प्शन ऑफ सोडियम एंड वाटर इट टेक्स प्लेस. कंडीशनल रीएब्जॉर्प्शन ऑफ वाटर एंड सोडियम. DCT is also capable of reabsorption of bicarbonate also. So reabsorption of bicarbonate ions also, and selective secretion of hydrogen and potassium ion. While the hydrogen and this potassium and hydrogen ion, they are selectively secreted into it. And ammonia also, to maintain the pH and sodium potassium balance in the blood. So in order to maintain the pH of the blood and the concentration of sodium and potassium ions, this secretion of hydrogen and potassium ions also takes place in the DCT. Now we will come to the collecting duct. Now what happens in the collecting duct, that I will discuss now. So the long duct which extends from the cortex of the kidney to the inner parts of the medulla. So this is the location of the collecting duct. Now the large amount of water, this could be this is here. The large amount of water, it is reabsorbed from this region to produce the concentrated urine. This segment allows passage of a small amount of urea into the medullary interstitium to keep up the osmolarity also. So small amount of that urea also it passes into the medullary region. Now it also plays a role in the maintenance of pH and ionic balance of the blood by selective secretion of hydrogen and potassium ions. So this is the role of collecting duct. So here this is the that role of the different parts of the nephron in the reabsorption of the substances as well as in the secretion of the substances. Now